Today, we are on site at the Fira Gran Via in Barcelona, Spain at the 2019 ESMO Congress. We'll be recapping some of the top news presented each day during the meeting, and soon we'll speak with both Drs. Suresh Ramalingam and David Spigel on some breakthrough studies in lung cancer. Welcome to OncLive News Network. I'm Gina Columbus. In non-small cell lung cancer, frontline treatment with osimertinib improved median overall survival by 6.8 months compared with erlotinib or gefitinib for patients with metastatic EGFR mutant disease, despite crossover between arms, according to eagerly awaited updated findings from the Phase 3 FLORA study. The median OS in the osimertinib arm was 38.6 months compared with 31.8 months with erlotinib or gefitinib, representing a 20% reduction in the risk of death with the third-generation EGFR inhibitor. At the final data cutoff for the study, 54% of patients remained alive at 36 months in the osimertinib arm, compared with 44% in the erlotinib or gefitinib group. Also in lung cancer, a final analysis of Part 1 of the Phase 3 CheckMate 227 trial showed that the first-line combination of nivolumab and ipilimumab led to a clinically meaningful improvement in overall survival compared with chemotherapy in patients with advanced non-small cell lung cancer, regardless of PD-L1 expression status. In a cohort of patients with PD-L1 expression of at least 1%, the median OS with nivolumab and ipilimumab compared with chemotherapy was 17.1 months and 14.9 months respectively. The median OS was 17.1 months with the combination and 13.9 months with chemotherapy in patients regardless of PD-L1 expression status. Frontline maintenance therapy with the combination of olaparib and bevacizumab improved the median progression-free survival by 5.5 months versus bevacizumab and placebo for patients with newly diagnosed advanced ovarian cancer following prior platinum-based chemotherapy plus bevacizumab, as seen in findings from the Phase 3 PAOLA-1 trial. Results showed that the median PFS with olaparib was 22.1 months versus 16.6 months in the placebo arm, representing a 41% reduction in the risk of progression or death with the addition of the PARP inhibitor. The benefit was more pronounced for those with tumors positive for BRCA1 or 2 mutations and for those with a homologous recombination deficiency score of 42 or higher by the MyChoice HRD Plus assay. Also in ovarian cancer, data from the Phase 3 PRIMA study showed that frontline maintenance therapy with niraparib had a median PFS of 13.8 months compared with 8.2 months with placebo in patients with newly diagnosed advanced disease who responded to platinum-based chemotherapy. The benefit was associated with a 38% reduction in the risk of progression or death with the addition of the PARP inhibitor. The median PFS was 21.9 months with niraparib compared with 10.4 months for placebo in patients with homologous recombination deficiency positive tumors. Results of the Phase 3 VELIA study showed that the frontline combination of veliparib, carboplatin, and paclitaxel, followed by maintenance veliparib monotherapy, led to a 32% reduction in the risk of progression or death compared with placebo plus chemotherapy with placebo maintenance for patients with high-grade serous ovarian cancer. In the overall population, median progression-free survival with veliparib was 23.5 months compared with 17.3 months with placebo. In those with BRCA mutations, the median PFS was 34.7 months versus 22.0 months for veliparib and placebo, respectively. In renal cell carcinoma, adding telaglenastat to everolimus extends progression-free survival versus everolimus alone in patients with heavily pretreated advanced disease, according to data from the Phase 2 ENTRADA study. Findings showed the median PFS was 3.8 months in the telaglenastat plus everolimus arm, compared with 1.9 months in the placebo plus everolimus arm. Sacituzumab govitecan demonstrated clinical activity with an overall response rate of 29% in patients with heavily pretreated metastatic urothelial carcinoma, according to findings of a cohort from the single-arm Phase 2 TROPHY-U-01 trial. The ORR comprised 2 complete responses, 6 partial responses, and 2 additional PRs pending confirmation. Sacituzumab govitecan was also found to be well tolerated with a manageable and predictable safety profile. In breast cancer, data from the MONARCHER trial showed that the combination of abemaciclib plus trastuzumab with fulvestrant improved PFS compared with trastuzumab and chemotherapy in women with hormone receptor positive, HER2 positive advanced breast cancer. Moreover, safety data was similar to the known safety profile of abemaciclib. Finally, more than 70% of patients with cisplatin-ineligible locally advanced urothelial cancer had objective responses to enfortumab vedotin plus pembrolizumab in a preliminary clinical evaluation. Responses to enfortumab vedotin and pembrolizumab occurred quickly and irrespective of PD-L1 expression status. The combination safety profile appeared to be manageable and tolerable and was similar to previous trials of single-agent enfortumab vedotin. We're here with Dr. Suresh Ramalingam, who's the Deputy Director of Winship Cancer Institute at Emory University School of Medicine, who's going to discuss some of the exciting lung cancer abstracts being presented this year. Thank you so much for dropping by. We really appreciate it.
Thank you for having me, Gina.
So, everybody's talking about FLORA. It's definitely a very exciting trial. It has been from the beginning, and now we've been eagerly awaiting the overall survival data that was presented at this year's meeting. So can you give a little bit of background on FLORA and talk about the OS data that is presented?
Absolutely. So the FLORA study was designed to compare the efficacy of osimertinib to either erlotinib or gefitinib in EGFR mutated patients. We enrolled patients with exon 19 and 21 mutations. These were previously untreated patients, so it's a clean first-line therapy trial, one-to-one randomization, osimertinib or erlotinib or gefitinib. And the primary endpoint was progression-free survival. Noteworthy for the survival analysis is that we allowed crossover. So patients treated with erlotinib or gefitinib at the time of progression, if they had T790, were allowed to cross over and get osimertinib. And we actually had a high degree of crossover in the trial. We've already reported the PFS results in the New England Journal of Medicine. Osimertinib resulted in a substantially improved PFS, 18.9 versus 10.2 months. The drug is approved in many parts of the world, including the US, for frontline therapy. Now we have the mature overall survival data, which is what we presented at this meeting.
Congratulations, it's a very exciting study.
Thank you.
How do these data kind of impact now your sequencing strategies, and what are you doing for patients who progress on osimertinib?
Well, first of all, what we learned at this session is that osimertinib improves overall survival. The median survival with osimertinib was 38.6 months, and in the control group it was 31.8 months. So it's approximately a 7-month improvement in overall survival that was statistically significant, and we can say it's clinically significant as well. We also noted that 47% of the patients in the control group who went on to receive some post-study therapy got osimertinib, which reflects the real-world situation. So clearly, this study met its primary endpoint and secondary endpoint. The question is, some people have said we can give first-line EGFR TKI with erlotinib or gefitinib, and then when they progress, you can sequence them. I think FLORA argues strongly against that. Number one, 30% of the patients in both arms of the study did not see a single post-study treatment after progression. That's because vast majority of them died before they could get another treatment. So for those 30% of the patients, there was only one line of therapy. And that argues for using your best agent first. The second point I would make is not everybody who progresses is going to have the T790M mutation. So if you start off with 100 patients with EGFR mutated patients, and if you all gave them gefitinib, only 1 out of 3 realistically has a chance of getting second-line osimertinib, and the other 2 out of 3 patients are going to miss out. So our argument is that this trial clearly addresses the sequencing issue, and with the sequencing in place, there's a survival benefit for using osimertinib in the frontline setting, and that's why we believe should be the standard of care.
Right. Thank you so much for highlighting all of that. So what's next for osimertinib? Where do you see this agent moving forward in the next couple of years in EGFR disease?
Right. What osimertinib has shown is it's effective, it is safe, and more importantly, it's active against brain metastases. All are 3 important characteristics when you choose first-line therapy. As we look at therapies that the patients will be getting for now years, not months, it is important that it's tolerated well and it doesn't interfere with their quality of life. So osimertinib meets those criteria. Obviously, we're not satisfied with where we are. We need to get better for our patients. We are already working on combinations with osimertinib to improve the outcome, to delay emergence of resistance to osimertinib. We're beginning to understand the mechanisms of resistance to osimertinib so we can develop informed combination therapies. And we're also moving osimertinib to earlier stages of the disease. There's an adjuvant therapy trial in resected lung cancer patients called ADORA that's already completed accrual, and we hope to have the results in the upcoming years. We're doing a study in locally advanced stage 3 lung cancer where patients get chemo-RT and then are randomized to osimertinib or not in the post-chemo-RT setting. So The drug is being tested in earlier stages of the disease. In metastatic disease, our efforts are focused on delaying the emergence of resistance and improving the duration for which patients benefit from targeted therapy.
Wow, those are some great points, and those sound like really interesting studies too.
Yes.
Great, thank you. So moving away from Flora, can you talk a little bit about the BFAST study as well as another trial that's being discussed here?
As we talk about targeted therapies in lung cancer, it's critical that every patient with non-squamous lung cancer get tested for these mutations. Now, we use tissue for testing, but increasingly plasma samples are being studied, and we know that plasma samples can pick up these common mutations. We also know that if you pick up a mutation by plasma sample, you can confidently treat those patients with inhibitors for those specific targets like EGFR, ALK, and so forth. The BFAST study was specifically looking at plasma sampling to see if they can pick up ALK. And what Dr. Gadgil and colleagues reported is a cohort of over 2,000 patients who were screened for ALK in the plasma, and they found about 5.4% prevalence in these patients. And based on that, they gave them alectinib, and the response rate was 87%, which we know alectinib is capable of doing in the frontline setting for ALK patients. So what the BFAST study shows is if you detect ALK by plasma, you can confidently treat those patients with alectinib and hope to see the same kind of benefits you would see if the tissue was positive for ALK. And I think we're going to be increasingly incorporating plasma in our workup because not all patients have sufficient tissue to do the testing. And also, there are certain clinical situations where the tissue is just not available.
Good, thank you. Now, what about some other lung cancer abstracts that are being presented this year that you think have some practice-changing potential?
Well, immunotherapy is an area where there's a lot of excitement and progress we've made. And I would point out two abstracts that were presented at this meeting. The one in the presidential session yesterday is the Checkmate 227 trial. It also came along with the parallel New England Journal publication. What we learned in this study is the combination of EP and Nivo in lung cancer is associated with improvement in overall survival in frontline therapy. This is the first evidence that EP-Nivo combination improves survival in non-small cell lung cancer. The study is a large trial. Over 1,500 patients were enrolled. We found that when you compare chemo to EP-Nivo in both PD-L1 positive and PD-L1 negative patients, EP-Nivo combination has superior survival. What is even more exciting to me is the duration of response within EP-Nivo stands out when you compare to chemo plus immunotherapy approach. So the ability that this regimen can sustain a response for years is something that I think will help us think through where this fits into the treatment landscape. The tolerability profile was also good, and there were no safety signals with the EP-Nivo regimen. We have to keep in mind that the EP that we use for lung cancer is a lower dose than what's used for melanoma.
Right.
So with this attenuated schedule, this is a well-tolerated, effective regimen. Now it should belong in the treatment landscape in the frontline treatment of non-small cell lung cancer. So that's clearly an exciting advance that we saw. Another study we learned about is the Empower 110 trial, which compared atezolizumab head-to-head with chemotherapy in PD-L1 positive patients. And in that trial, we learned that if you had a patient with very high levels of PD-L1 expression, atezo was associated with a favorable overall survival compared to chemo. The intermediate and low PD-L1 expression patients did not have as significant a benefit. So these results are not very different from what we've seen with Pembro versus chemo, where the highest PD-L1 positive patients had the survival advantage with Pembro monotherapy. So this trial, Empower 110, tells me that atezo can be used in the frontline setting when it receives the approval as monotherapy for those highest PD-L1 positive patients.
Great, thank you. It seems like a little bit more of a trend this year with some chemotherapy-free regimens across different subtypes, right?
Absolutely. And obviously, it'll boil down to what else can we do to increase the proportion of patients that benefit from immunotherapy. What we see is a subset of patients benefit, and they have sustained benefit, and you see the tail of the curve getting better. But we need to see how to improve the proportion of patients who get benefit. So novel combination approaches, looking into biomarkers, are going to help us deliver that next set of progress.
Well, great. Thank you so much, Dr. Ramalingam. This was amazing. Thank you so much for sharing your insight.
Thank you for having me.
Yeah, of course.
Here now to expand on some of the breakthrough studies with immunotherapy in lung cancer is Dr. David Spigel, who's the Chief Scientific Officer and Lung Cancer Program Director at Sarah Cannon Research Institute. Hi, Dr. Spiegel. Thank you so much for being here today.
Hey, Gina. Thanks for having me.
Oh, sure. Of course. So let's first touch upon the phase 3 Empower 110 trial.
Sure.
So this is looking at single agent atezolizumab versus chemo in the frontline setting for wild type non-small cell lung cancer with some other certain disease characteristics in there. So you could touch upon that trial?
Yeah, I think over the last 4 or 5 years, as immunotherapy has found its way into non-small cell lung cancer treatment, the goal is, can you get it into earlier lines of therapy? We know from Martin Reck's pivotal Keynote 024 study that pembrolizumab found its way into first-line treatment in high PD-1 expressing patients. At this meeting, we've heard about Checkmate 227, which we may talk about here in a moment. And then another big study in the first-line space, as you say, was this Empower 110 study. So really simple design. Could you take a PD-L1 inhibitor, in this case atezolizumab, and take on platinum doublet chemotherapy in newly diagnosed patients? A couple of caveats about this study. It was a randomized phase 3 with an OS endpoint, but you had to have PD-L1 expression, as you say, to get on this, but the assay was a little different than the Merck studies. So this was the SP142 assay, which measures PD-L1 expression by what's called TC, or tumor cell expression, or IC, immune infiltrating expression. And to get on the study, you could have any expression as long as there were some, so 1+ through 3+. The endpoints, it's worth taking a moment to understand this. So the endpoints were designed to be looked at in a hierarchical design. So you start with the 3+ and then go to the 2 3s, then the 1 2 3s. And you have to hit all of those to then look at the secondary endpoint of PFS. So I'll just jump into it. About 540 patients were randomized. About 35% roughly had the 3+ group. That's about 100 in each arm. And in that group, atezolizumab was superior to platinum doublet chemotherapy, regardless of histology, so squamous or non-squamous. The overall survival was in favor of atezolizumab. The hazard ratio showed about a 41% reduction in the risk of death if you're on atezo. So pretty substantial findings. The study met its endpoint. When you look at the 2 3+ survival advantage, it was in favor of atezo, but the p-value was 0.04. And 0.0416, I apologize.
Okay.
Because of the design, you had to hit 0.04, so it just missed it statistically. And therefore, the 1 2 3s, which also show a trend favoring atezo, technically is not positive either. When you go to PFS, it's kind of the same story. The PFS looks substantially in favor, about almost a 40% reduction in progression or death with atezo, but because you didn't hit the 3 strata, you can't say that's statistically positive. Response rate, duration of response appear to be in favor of atezo as well. The safety profile looks very good. And so what do I think this means? I think this means we have a new standard of care in the first line setting in addition to pembrolizumab, and as we'll talk about in a moment, nivolumab, ipilimumab, hopefully in the 3+. And there's ongoing work to, this was an interim analysis to see if another group hits, so there's a chance 2 and 3s or 1 2 and 3s can hit. And there's ongoing biomarker work, so because 22C3 is an assay that's probably more prevalent than the SP142, there are going to be analyses presented here soon showing how the data look when you look at it using a 22C3 assay or an SP263 assay, and also some blood TMB data are going to come out. So more to come, but this is, I think, a big step because it brings another therapy into the first line setting.
Definitely. It's a study with a lot of layers in it, too.
A lot of layers.
Yeah.
Gosh, every study now seems to have multiple layers and endpoints, so that's just the way it is.
Yes, speaking of which, let's move into the Checkmate 227 trial. Again, another trial with a lot of pieces to it.
Yeah, it's funny because I live in lung cancer, as do a lot of my colleagues, and you got to keep up with it. So I'll try to make this simple. So Checkmate 227 was basically the attempt to see how BMS can get into the first line space. You may remember a pivotal study of Nivo versus platinum doublet chemotherapy in the first line called the Checkmate 026 study was negative. and so that didn't mirror what we saw in Keynote-024 with pembrolizumab. So this study's a little different. It was nivolumab and ipilimumab, a regimen which already has a U.S. label in 3 cancers, so in advanced renal, advanced melanoma, and advanced MS-high colorectal. So we know it's an active regimen. We've known it's active in small cell and non-small cell, but this was the pivotal registration trial to get it approved. Complicated design. Nivo-Ipi, chemo-Nivo, or chemo. There was no Nivo arm alone because of the 026 data. Everything was looked at. PD-L1 expression, histology, squamous, non-squamous, and TMB. And I think, Gina, what you're alluding to is a pivotal New England publication from Matt Hellman, and many presentations have focused on TMB. That is to say that Nivo-Ipi has looked great as a regimen, especially in the high TMB population, and I think we all thought that's the path to registration. And at this meeting at ESMO 2019, we got a press release ahead of time, but we saw the data that Nivo-Ipi was better than platinum chemotherapy. However, not because of TMB. It actually looks okay with the TMB group, but in the PD-L1 1+ or higher. So here we are now back to PD-L1. And what I think this means is that Nivo-Ipi will become another first-line regimen available in our expressors. And so in my view, we have 3 regimens kind of showing the same thing, that if you have expression, in the case of Atezo, high expression, and you can make the argument in the case of Pembro, the highest expressors kind of carry the weight, even though it has an approval in the 1% or higher. We have a 3rd regimen available. A little bit more safety concern with that, with Nivo-Ipi, and I think if you haven't used it, that'll be something you have to get used to. But I think it's good for patients to have different regimens, and hopefully this provides new platforms to build upon.
You definitely simplified it.
Yeah, right.
Yeah. So let's move on into small cell lung cancer. So
yeah, sure.
There's a presentation of updated OS analysis of the Empower 133 trial with atezolizumab and some chemotherapy.
Yeah.
So what are your thoughts on these data?
So, small cell has been obviously something I'm very interested in. We see a lot in our region in the southeast of the United States. Chemotherapy is good, it's just not great. It doesn't have lasting effects in everybody. Everybody recurs. And our options beyond that are not great. And so what we learned with Empower 133, and actually I may, with your permission, talk about the Caspian study.
Yep.
We have 2 studies that show if you give platinum etoposide with a checkpoint inhibitor, in the case of 133, the PD-L1 inhibitor atezolizumab, in the case of the Caspian study, the PD-L1 inhibitor durvalumab. Both studies, in my mind, show kind of the same thing, that there's clearly a survival advantage if you get the triplet as opposed to the standard of care for the last 20 to 30 years of platinum etoposide. And that's a big deal. We're a little spoiled on the non-small cell side because we've seen such great curves in survival and PFS and response rates. I think a lot of us, or all of us, were hoping that the results would be even more substantial than they have been. And that's not to say they're not good, and they're huge steps forward for our patients. I think we want more. We want to learn more about who benefits the most. Can you select out those patients? So far, PD-L1 and TMB don't seem to help at all. Does everybody need maintenance? Does everybody need to get that triplet? What about radiation to the chest? What about PCI? There's still a lot of questions. And once you relapse, are you done with IO? Kind of like in non-small cell, do you have a chance to get it again in some other way or some combination? These are the things we'll discover in the next probably 2 to 3 years or 5 years.
All right, great, thank you. So the BE-FIRST trial, that's a study looking at blood-based TMB as a predictive biomarker for frontline atezolizumab. So just kind of looking at trials like this, can you kind of speak to the importance of looking for kind of this ongoing search for biomarkers, and especially biomarkers that are predictive of response to immunotherapy?
Yeah, I think what is exciting about this kind of work is the idea that we're going to start using the blood to make decisions. I mean, that sounds kind of silly because we've always done that in medicine with blood work. But we're talking about more sophisticated blood tests now, genomic testing, and you're referring to something that's even more sophisticated, tumor mutation burden. But in the blood now, in the standard of care setting, you can measure things like EGFR. You can measure markers to help you guide therapy. In our network alone, we're doing about 700 comprehensive molecular tests a month, and half of those are blood-based, and we're seeing that grow exponentially. You know, that has me worried on the research side in that, is that enough? You know, hopefully we're getting tissue too, but I do believe that in the future, blood will probably be the way we're doing everything. And so BE-FIRST and a companion study called BE-FAST are designed to use blood up front to make decisions. Can, for example, a TMB value help you make a decision, in this case, who gets atezolizumab or platinum doublet chemotherapy? Can you also find RET? Can you find EGFR and implement decisions in care because based on that blood test that help the patient do the best? Blood tends to be faster in general. We get it back in 5 to 10 days. Tissue testing right now is taking about another week or two beyond that. And so this offers a real opportunity to make earlier decisions in care with less invasiveness. So I'm excited about where this can go and hope we're going to see more data, more studies like this.
That's great. Thank you so much.
Yeah, thanks.
What there's been a lot of great lung cancer data being presented here at ESMO. Is there any other studies focused on immunotherapy and lung cancer that you'd like to share light on?
Well, there's a lot. You know, I think the FLORA data, not immune-based topic, but certainly osimertinib's survival advantage over erlotinib and gefitinib is pretty nice to see. I actually was going to jump around a little bit and tell you that I'm pretty excited about the work in DNA repair with the PARP inhibitors in ovarian cancer and also in prostate cancer. I think the reason I'm excited about that is I think those are untapped places for lung cancer research. I mean, folks have studied these kind of drugs in combinations in small cell, but not in selected ways. You know, we saw this past year BRCA in pancreatic cancer can be important in making treatment decisions. And the role of DNA repair targeting and immunotherapy, I think, is the next kind of is a frontier that everyone is kind of excited about. What is the interplay? Are there ways to select patients based on blood testing or tissue testing or other assays? I think that's going to be a next exciting area for all of us, but especially in lung cancer. I think it's kind of an area that's ripe for some good research.
Thank you so much, Dr. Spigel.
Great.
This was fantastic. Really appreciate you coming by
okay,
today.
thanks, Gina.
Great.
Appreciate it.
That's all for today. Stay tuned for tomorrow's OncLive News Network On Location, where we will sit down with Drs. Hope Rugo and Joyce O'Shaughnessy on the key breast cancer abstracts, Dr. Tanios Bakai-Saab on practice-changing studies in gastrointestinal cancers, and Dr. Mansoor Mirza on the latest in ovarian cancer. Thank you for watching OncLive News Network. I'm Gina Columbus.